I've made a mistake. Good. So my lab, among other things, is one of the 3 labs that have been asked by the National Aging Institute to take suggestions for drugs that might extend lifespan in mice by slowing the aging process and to test them to see if they work. Most of them don't work, but a few of them do work, and that's one of the reasons that the work I'm presenting is often a way of getting potential ideas about how to slow aging and thereby retard disease in people. The first half of the talk brings you up to date on that. But once we have drugs that can slow the aging process, they're spectacularly useful tools to begin to ask questions about what are the mechanisms that slow aging, how can you discover new drugs, and how can you begin to make molecular attachments between the aging process and the diseases of aging. That's the second half of the talk, and it focuses on a new concept. I feel like I'm a televangelist marketing Jesus to you. What I'm marketing is aging rate indicators. And at the end of this hour, if you stay till the end, you'll see why I think that's a thrilling thing to be enthusiastic about. You have to press a button somewhere. Something bad has now happened. Try again. All right. So everybody knows you can slow aging in mice and rats and many other critters too by just giving them less food to eat than they want. We're not going to talk about that. And in 1996, Andrzej Bartke and Holly Brown-Borg and others published the first mammalian gene that extends lifespan in mice. It was about 40%. It's the Ames dwarf mice. There are now a half a dozen or more that can do that. We're not going to talk about that either, because none of us wants to stop eating breakfast, and because none of us wants to change our genes. What we want to do is take a pill, so that's what I'm going to talk about. The Interventions Testing Program, the ITP, has the following features. We use no inbred mice whatsoever. We always use genetically heterogeneous mice because we would like to discover things that don't work merely in black 6. We would like them to work in more or less all mice. So we have mice that have the same 4 grandparents, and in our population of by now more than 20,000 mice, they all have the same 4 grandparents. They're all brothers and sisters, but every one of them has a unique genotype, and we hope this will produce robust findings. Anyone, which includes everyone in this room, can suggest a drug. There's an annual call for suggestions. So all you have to do is write down a few pages. Here's a drug I think you should test. Here's why I think you should test it. Here's why I'm fairly sure it won't hurt mice, so your veterinarians can relax. And we'll pick 5 or 6 or 7 a year for you. They're tested, and this is expensive but important, at 3 independent sites: my lab at Michigan, Randy Strong's in Texas, and at the Jackson Lab by Dave Harrison and Nadia Rosenthal. Ron Korstanja is about to replace David as head of the Jackson Lab program. That means if the drug works at all 3 places, it probably will work for you too. That's an internal parallel replication. And we have lots and lots and lots and lots of mice. We have 300 male controls and 300 female controls each year, and half that number for each sex for each drug. The goal was to have 80% power to detect even changes of 10%. To give you a sense of whether 10% is a big number or a small number, if you have a drug that cures cancer and no one ever gets cancer again, average human lifespan goes up 3%. If you have a different drug that abolishes heart attacks and strokes and no one ever gets a heart attack or stroke again, human lifespan goes up 3%. So when you see drug effects that are 10%, you do that calculation, 10 divided by 3. This is about 3 times better in terms of percentage increase of healthy lifespan than a complete cure for cancer. 10% is a lot. Most drugs don't work. This was the hypothesis, and it was absolutely correct. If you ask smart scientists to come up with a drug that they think is going to slow aging in mice, they're wrong most of the time, and here are the first 27 that don't work. I've put into red all of the things that are famous. Curcumin doesn't work. Resveratrol certainly doesn't work. Resveratrol doesn't work over and over and over and over again. Nicotinamide riboside, which you can buy at $90 for a month's supply, that doesn't work. Fisetin, allegedly a senolytic, that doesn't work. Metformin didn't work. And there are lots of others substantially less famous. Some of them did work. The second one that worked, but the first one that people started to pay attention to was rapamycin. This is not the first paper, which was in 2019. This is the follow-up dose response curve paper in 2014. And you can see, looking at the females, it's dose dependent. You go up to 42 parts per million in food and you're getting 26% increase in the median lifespan. Importantly, the maximum lifespan, sort of the age at which the last 5 or 10 or 15 mice drop off, is also extended by an equivalent amount. It works in males too, and it's dose dependent in males. At a glance, you can see it sort of seems to work better in females, but that's an illusion. And it's because the doses in the blood of females are 3 times higher than the doses in the blood of mice. So if you give them the food that has the same concentration of the drug, the females have more of it in their blood. And if you adjust for blood concentrations, this sex differential at least gets smaller. These are the statistics. The median increase at the lowest dose went to 3, and 23% increase, which is more than 3%, was seen in the males at the higher dose, and in the females at the highest dose, it went up to 26%. This is just to emphasize the disparity between what we can easily do in mice and what could do in a fantasy world in which suddenly cancer is cured. That gives you an increase. The x-axis is a little confusing. That's how many remaining years of life a 50-year-old white American woman lives. So your average 50-year-old white American woman lives another 30 years. And if cure for cancer occurs, then she gets another 2.7, 2.6 years out of that. The same is true for heart attacks. And even for curing cancer and heart attacks. And I don't need to remind you, that's not going to happen, right? It's certainly not going to happen soon. It's never happened. They have no drug that does this even in mice, let alone in people. You can nibble away around the ages. But if there were a magic pill that stopped cancer, it's that blue bar, the Cure Cancer Today bar that you'd wind up in. This is what rapamycin does today in mice. It's dramatically more impressive than everything else being done at the NIH or in all the cancer centers in the world or in all the Alzheimer's centers in the world. I think we should study aging. That's my personal opinion. Okay, so is it merely extending lifespan? A lot of these mice die of cancer. Maybe it's a dynamite anti-cancer drug and it blocks every single kind of cancer. To a cancer biologist, that would be good news, but to an aging biologist, that would be terrible. I don't care about curing cancer. I just want to slow aging. So this is a pathology study done by J. Irby Wilkinson. Here, for instance, you can see this is these are 22-month-old mice. They were euthanized so we could get them while they were healthy and look at pathology. You see liver oops, oh no, mistake. Liver degeneration goes up with age, but not if they've had rapamycin. Myocardial nuclear atypia goes up with age, but not if they've had rapamycin. Adrenal tumors go up with age, but not if they've had rapamycin. This is tendon stretching. This is done by Sue Brooks and her grad student, Lauren Wood. Take a tendon out of the mouse, try to stretch it. That's the panel on the left, and then let it snap back. That's the panel on the right. Both of those change with age, just like your tendons get stiffer and stiffer with age. Rapamycin blocks both of those, both the stretchability and the resiliency, the elasticity. And here are 5 other lesions that Irby was able to detect. 4 of the 5 show rapamycin inhibition. So the conclusion from this is that rapamycin is not merely extending lifespan, but it is slowing down a wide range of age-associated changes. We've done many more than these. These are the ones that came out of the pathology study. The take-home message I want people to understand is there is now, I don't want to be disparaging, a puzzling thought that you can either have lifespan or health span. About twice a week, someone says to me, I'm not interested in increasing lifespan, I'm for health span, as though you got one or the other, as though there were a trade-off. And that's a very bad way to think about this. The reason these drugs extend lifespan is because they extend health span. The reason these animals are living such a long time is that they stay healthy for a long time. In principle, one can imagine a drug that makes you live forever, but you're sick and sick and sick and sick and sick, just like in the Dorian Gray fantasy. People wouldn't want to take that drug, but more importantly, we have no evidence that such a drug exists. All of the drugs that we've developed, all of the ones we've studied extensively, don't do that. What they do is, not too surprisingly, they extend lifespan by keeping you healthy for a longer period of time. You should tell this to someone who says, I don't want to extend lifespan. All I want is to stay healthy. Now, it turns out, and this is a big surprise, and this is why the paper got into Nature, I think, rapamycin actually works even if you start in old mice. The median survival is about 26, 28 months. We thought that if you started at 9 months of age, you'd get a big effect. If you started late at 20 months of age, you might get no effect. You might get a really small effect, and we were wrong. It works just as well if you start at 20 months of age than if you start at 9. Something bad is still going on when you're a 20-month-old mouse. In terms of percent lifespan completed, it's like a 55 or 60-year-old person that rapamycin inhibits, and which has a profound, long-lasting effect on your survival. The second of the drugs that we've published is acarbose. Acarbose is a frontline therapy for adult diabetes, particularly in countries, as for instance, Asian countries, in which they eat a lot of rice, but they don't eat much wheat. If you take acarbose and you have a lot of rice, it doesn't hurt your digestive tract, but it makes you feel bloated if wheat is your main carb source. What it does is it stays in your gut. It doesn't go into the body really, and it blocks the bacterial enzyme that digests starches to sugar. So if you have a couple of bagels, the starches in a normal person, not in acarbose, rapidly get degraded to sugars. The sugar's absorbed, and you get a blood sugar peak after you eat my third donut for the morning, something like that. Acarbose blocks that enzyme, and so the carbs are very slowly digested. You don't get a big glucose spike. The glucose goes up, but it goes up a little bit, stays up for a long time because it takes you a long time to digest the carbohydrates. That's why it's so good for diabetes. And here's what we saw. David Allison and Dan Smith suggested this. Acarbose gives a significant effect in both males and females, but it's always bigger in males. A theme we'll come back to, and I don't understand why that is. No one understands why that is. In human treatment, it works in males and females to help diabetics, but although it's significant, and we've done it now 3 times, it's always significant in males and females. It's also always bigger in the male mice. 22% lifespan extension. That's quite similar to the lifespan extension we see with our initially tested dose of rapamycin. Now, if you start it in midlife, it works half as well. It's a little different from rapamycin, which works just as well if you start late. For acarbose, you do get an effect if you start in midlife. It's not as big. It's still significant. And I don't have time to walk you through the details, but we've looked a lot at health span types of things. Mariana Nagurski looked at inflammation of the brain, and in males only, acarbose blunts that. Charlene Day looked at changes in ventricular size, left ventricular mass, and in males only, acarbose blunts that. Mike Garrett looked at age-associated changes in glucose homeostasis, and in males only, acarbose blocks that. And of course, cancer, which is the thing that kills them. I mean, I've been trying for 15 years now to talk to cancer biologists at Michigan into pretending that I'm an anti-cancer biologist. Unlike them, we have things that actually stop cancer, and they are unwilling to think about it because I'm not an official card-carrying cancer biologist. We have drugs, I'll show you 4 of them, that stop cancer dramatically. It reduced the age-associated cancer, age-adjusted cancer incidence by 10-fold, but it emerged from its aging research, and so the cancer biologists have no interest in that. My understanding is that Memphis has some cancer biologists, and if you happen to bump into any of them, you might want to mention to them that if they're willing to put aside their pride and talk to an aging biologist, we have anti-cancer drugs. They're just in a little bottle that says anti-aging drug. Now, what happens, this is the most recent data, what happens if you combine rapamycin plus acarbose? And the answer is that for females, remember in females, acarbose doesn't work very much. The answer is you get the same exact effect as if you use rapamycin by itself. Acarbose doesn't hurt the females, it doesn't help them. But in males, you get biggest effect we've ever seen. This is a 29% increase in median survival when both drugs are used together. It's the biggest effect percentage-wise we've ever seen, a 29% increase in median longevity. And also it's the first example we've ever seen of 2 drugs, which when used together, give you a bigger effect than either drug used alone. We're certainly now trying in the mouse colony. We have lots of other drug combinations, but this is the first of them to reach publication. The third drug I want to talk about is 17-alpha-estradiol. 17 beta estradiol is famous. It's called estrogen. This is not estrogen. This is the same steroid, but one of the hydroxyl groups is tilted out of the plane. It's in a different plane. It's a stereoisomer. And because of that shift in that one group, it's alpha estradiol. Because of that shift, it is much lower, probably tenfold lower, in its ability to stimulate the famous estrogen receptors, the one that plays such a role in secondary sexual characteristics and in breast cancer biology, et cetera. So it was given to us by someone who said, I'll bet you could get male mice to live as long as female mice, and you wouldn't turn them into girls. No one wants to take a drug that will turn them into a girl. So why don't you give this to the mice, and the males will just bump right up to the same survival characteristics of the female mice. Jim Simpkins said this. And to my pleasant surprise, it did much better than that. The male mice treated with 17-alpha estradiol not only do as well as the females, they do much better than the females. The drug has no effect on the females, but it has a really big effect on the males, so that the drug-treated males have a longer survival than the females, whether the females have been treated with drugs or not. We'll come back in the last half of the talk to cellular and biochemical changes that are sex-specific, which give us some clues as to what might be going on there.
Does animals produce this like
No, or not much. It's a little obscure. There's one paper that says the brain makes some of it, but it's 10 years old. I've never seen it replicated. What if you start 17-alpha estradiol late? It works great in males, right? So here we're starting at 16 months or at 20 months of age, and the benefit you get is still present, even if you start as late as 20 months, although starting at 16 months is better. Now, sex-specific physiological changes are shown in this slide. This is also Mike Garrett's work. You can see grip strength in the top two panels, rotor rod that is sort of balance and motivation on a spinning rod. The young and old differ, but in both sexes. As you get older, you get worse at grip and you get worse at rotor rod. In the males, if they've been on 17-alpha estradiol, they're as good as young. It has preserved this aspect of their physiology as well as their lifespan. And in the females, it does not do that. It does not allow them to retain youthful characteristics on these two different fitness measures. We're about to start a test where cognition is the endpoint. I will be eager to see how that comes out. So this is late start. The same graph I just showed you, except these mice didn't get the 17-alpha all their lives. They got it starting at 16 months of age. This is Mike Garrett's work as well. And the answer is essentially the same thing. There's a change with age, and even starting this stuff at 16 months of age, you still, at 22 months of age, after only a mere 6 months of exposure, you now have either gone back to youthful characteristics or at least failed to show aging from 16 to 22 months. And I don't have time, all this is published, and so I won't have time to work you through it. But for instance, Mariana also looked at hypothalamic inflammation, and that too, in a male-specific way, was prevented or possibly reversed, we don't know, by 17-alpha estradiol. And of course, cancer. If any of you are cancer biologists, I can give you some of this anti-cancer drug, and you can prevent cancer if that's the sort of thing you might want to do. The last of the published drugs is canagliflozin. Canagliflozin is transforming treatment of adult onset diabetes. I take it, for instance, because I was pre-diabetic. Doctors give it to diabetics and pre-diabetics. What it does is it goes to the kidney, and it fiddles with the kidney's ability to lose glucose in the urine stream. So when you've had my 3-donut breakfast, if you're a normal person and you have a 3-donut breakfast, your blood glucose goes way up. But if the kidney pump, SGLT2, has been poisoned by canagliflozin, then all of that glucose goes out in the urine and you don't get that big peak of glucose. So we wanted to test this because we wanted to be darn sure that our theory, acarbose is working by avoiding peak glucose, was correct. This is a different way of blunting peak daily glucose, but not working in the gut, this time working in the kidney. And as you can see, just like acarbose, it's dynamite for males. And like acarbose works for females, but not as well. And canagliflozin doesn't work for females at all. So this implies that peak glucose levels are important for aging and, of course, also for cancer for reasons that are completely not understood. So here's a pathology study. It was published by Jessica Snyder and Warren Ladigas. They took tissues from 22-month-old mice, males and females, treated with canagliflozin. And you can see, these are all males here. Here's some lesions, adrenal cervical neoplasm, arteriosclerosis, cardiomyopathy, glomerulonephropathy, hepatic change, lipidosis, pancreatic atrophy. And the canagliflozin treated mice have less of all of those if they are male. So canagliflozin in males is blunting age-associated changes in all 6 of these tissues. But in the female, only one, the pancreatic atrophy. And this is not the beta cells, this is the enzyme secreting cells of the pancreas that are making digestive enzymes and putting them into the gut. The other 2 are the only other 2 significant things, but they are not seen in, they're barely significant and like 0.09. And this is one in which the drug is actually causing the disease rather than preventing it. So the take-home message here is that age-associated pathological changes in at least 6 organs are blunted by canagliflozin, but in males only. What if you start canagliflozin at late age? So this is an incomplete experiment. I'm allowed to show you data only if half of the mice have died. Okay, so this is now legal. I'm allowed to show this. We'll publish it in a year or so. And you can see that even starting at 16 months of age, in male mice, canagliflozin is still having an effect. It's not something you have to take your entire life. In females, surprisingly, it's harmful if you start at 16 months of age. Now, we have a clue about this because we've actually measured canagliflozin in the blood of males and females. When they are young, canagliflozin is 3 times higher in female blood than in male blood. When you get old, everybody goes up an additional power of 3. So by the time the females are old, their blood canagliflozin concentrations are 9 times the levels that canagliflozin produced in young males. And our hunch is that that's toxic. Our hunch is that that's what's causing this harm to females. They have just so much canagliflozin that it's really not good for them. So we're going to try a couple of obvious things. We're going to try females at a lower dose, right? We're going to try females where we give them the drug, but only till 16 months of age and then never again. We're hoping in this way to find a treatment regime for canagliflozin that will work in females, but we're just getting started on that. So this is just for the record. We have other drugs that work too. These are the published ones. NDGA, which is nordihydroguaretic acid. If you've ever been on a Native American campground and you've been fed a tea made from the creosote bush, this is the active ingredient, NDGA. It's an antioxidant. It's also an anti-inflammatory. You can get it in Europe as an acne cream, believe it or not. It's not available. It's not FDA approved in the United States. But we've done 2 experiments now, and in both cases, it gave a 12% increase in males. It really works in males, but we had bigger, stronger effects from other drugs, so we're not following it up. It's also quite expensive. Protandim is a NRF2 activator. It gave a 7% increase in males, significant, but not large enough for us really to follow it up. Glycine, we published this in 2019. If you dump a ton of glycine into the food, 8% of the weight of the food is glycine, you do get increased survival. You get it in males and females. It's significant. All 3 sites, it's significant in both sexes, but it's really small. It's only a 4% or 5% increase. And so once again, we're not following up on that. We published an early paper saying, hey, aspirin is good for you, but we were unable to replicate it, and that's why that's crossed out here. Okay, so here, this is the list of the drugs that are now under evaluation. This is not top secret. In fact, it's posted on the NIA website. All the drugs we've ever tested, including those for which we do not have published data, are posted on the NIA website. So if you want to propose a drug and you're interested in asking whether we've already tested it, that's the place to look. Some of these are in red. And that's because we are about to publish, as soon as we complete the website submission form, Two new drugs, astaxanthin and meclizine, which extend lifespan, not very much, 8 to 12%. And again, only in males for reasons we do not understand. But the interesting about them is that they're available at the local Target or CVS or Walgreens or Walmart. They are available without a prescription. There are a lot of people that meclizine is used for seasickness, and astaxanthin is used because people think it might be good for you, and they didn't bother to ask the FDA. So you can buy this stuff right now, should you choose that. I don't know that they are known to be safe, taking a state daily for 70 years. I have no opinion about that. I rather doubt it. But for mice, they extend mouse lifespan. The 2020 cohort is the one that's going to really, I think, be exciting. It's the late start canagliflozin, and you've seen that data already. Sodium thiosulfate, there was a scientist named Jay Mitchell who died, unfortunately, in a road accident in Switzerland. And Jay believed that hydrogen sulfide was a key element in lifespan and aging control. So he told us test sodium thiosulfide, and that is giving a positive result. And the last one is 17-alpha hydroxyestriol. It's not estradiol, it's hydroxyestriol, and that is giving a huge effect in males. But again, to our frustration, no effect in females. So this is a summary of the key results. We are in terms of our tools to control aging, we're here, right? These are Stone Age choppers and cutters and things. We've got tools. We can do things with tools. These are our tools. We hope to have this kind of tool somewhere down the road, a decade or 3 decades from now. You can see that this table shows the median lifespan change. Most of these have been replicated. Canagliflozin now has been replicated. And you can see a couple that work in females, 3 of them, and 5 of them that, 6 of them that work in males. So that's the summary of where we are so far, with more in the pipeline. I'm going to leave this part of the talk and spend the last half hour. You have a question.
Yes. So how do you dose?
We put it in the food.
How do you know how much the animal is taking?
A mouse eats about 5 grams. A bigger mouse eats 6 grams. A littler mouse eats 4 grams.
So a mouse will take exactly the amount.
No. Some mice eat more, some mice eat less.
So you actually don't know.
We know pretty close. I'm sorry you're so disappointed. We know that the mice are getting enough of the drug that it's actually increasing their lifespan very dramatically, and that's why we're happy. We feel that knowing whether a mouse ate 4 or 4.2 grams of food is not going to help us solve that problem.
Can you guess what the department is?
No. So I'm going to talk about something entirely different now. Yes.
About the section, because the sections
No.
are very different.
They sure are.
Yeah, you want to take questions?
I'd prefer to go through this other material if that's okay, and then we can have questions on both sets whenever you're ready. You think that's a bad idea? What is your question? Okay, then let me talk about aging rate indicators. So this is a new theme, something that people haven't talked about. So the problem is it's not really easy to guess what drugs might extend lifespan and slow aging in mice. The challenge for this is to find a fairly quick way of improving the frequency of good guesses. So here's our approach. We have 10 different kinds of slow aging mice. There are 5 single gene mutants. I'll show you the details in a moment. We have 4 drugs, and we have the calorie restriction diet. We want to ask, what do they have in common as young adults? Is there anything that is seen in all 10 kinds of those mice as young adults? Because even though the slow aging mouse was produced by different mutants, different drugs, different enzyme targets, dietary change, if there's something they have in common, the thing they have in common, the shared mechanism, might be the key ways both to teach us what is upstream actually controlling the rate of aging. That's why we call these aging rate indicators, and they're clues to downstream diseases like cancer and diabetes and Alzheimer's and famous diseases. So the bonus for this study, not only are we hoping to find a screening test that we can quickly do, quickly being 2 months, quickly do on mice with a drug to see if the drug is going to work, but also as a bonus, we're going to get shared mechanisms to postpone age And, you know, if you're a geneticist, say, you might want to map QTLs, that quantitative proteolysis that control those things. Okay, here's the panel of test objects. We have 4 published and 1 just unpublished, but we've just submitted the paper. The Amesdorf, Snelldorf, PAPA knockout, and growth hormone receptor knockout, they all live 20 to 40% longer. P10 is the famous tumor suppressor molecule. It's a lipid and protein phosphatase. It's a key tumor suppressor in people. So tumors knock it out. That turns out to be a long-lived mouse. Others found that and published it. It turns out also to show all the aging rate indicators that I'll show you in a moment. 1 diet, calorie restriction, and the 4 drugs I've just introduced you to. So the goal here is to find changes induced in young adults by all of these long-lived mice. So let me give you some examples. The data are almost everything I'll show you here is published, except for the P10 stuff. So this is not everything. This is illustrative examples. So the first of these I want to talk about is a protein called GPLD1. GPLD1 is thought to be very important because if you exercise, it goes way up in your plasma. If you're human or a mouse, exercise causes GPLD1 to go up. And another lab has shown that if you artificially increase GPLD1 in mice, cognitive function increases. People have a pretty good idea that maybe exercise is good for cognitive function. And it's not a bad theory that GPLD1 might be a mediator or maybe even the mediator of exercise transition to cognitive increases. So with that in mind, what Gina Lee did was measure GPLD1 as a protein in the plasma of 2 long-lived mice. She's actually done it in 5 long-lived mutant mice. I'll show you just the first 2, the Snelldorf and the Gerkel mice. And you can see in both males and females, the dwarf have substantially more plasma GPLD1. Every one of these has a little statistics thingy here. And it's the genotype line that tells you whether there's significant difference between the 2 genotypes. Now let's look at drugs and the calorie restriction mouse. And the controls, that's wild type. These are untreated animals. Here's canagliflozin, and look, GPLD1 is up in the plasma. Calorie restriction, look, it's up in the plasma. Acarbose in both sexes, interestingly, it's up in the plasma. 17-alpha estradiol, it's up, and rapamycin, it's up. And P10, it turns out to be up also. So for all 10 kinds of slow aging mice, GPLD1 is up in the liver and in the plasma at the protein level. So I didn't show you the Ames mutant, I didn't show you P10, and I didn't show you liver and fat because the graphics look exactly like this. Only the name of the tissue has been changed. Now let's look at the brain, because it's thought that GPLD1 goes to the brain and improves cognitive function. And in fact, the lab that first demonstrated that looked at BDNF, which is brain-derived neurotrophic factor, and that is thought to protect brain cells from damage and from stress. So what Gina did was she measured in the hippocampus both BDNF and double cortin, DCX. Double cortin is a sign of neurogenesis when neurons are produced, when stem cells in the brain are producing new neurons, double cortin goes up. It's considered an indicator for production of new neurons. And you can see double cortin shown here and BDNF not shown here, but it's in the published paper, goes up. It goes up in the Snell-Dwarf mice, in the growth hormone receptor knockout mice, in the Amesdorf mice, in P10, and in PAPA. And you can see that it goes up for all 4 drugs and for the calorie restriction diet. So it is one of those shared factors shared amongst 10 different kinds of slow aging mice. We predicted that, of course, because GPLD1 is supposed to do that, but it's nice to see that was correct. Now let's look at the fat. UCP1, excuse me, is uncoupling protein 1, and elevation of it helps keep you lean, prevents obesity, and allows mitochondria to turn fuel into heat rather than into stored fat. So it turns out that we've evaluated three fat depots, the brown fat, the inguinal white fat, which is subcutaneous, and the perigonadal white fat, which is intra-abdominal fat. And for the Gurkha mouse and the Snell-Dwarf mouse and the CR mice and 3 of the 4 drugs, we see the same thing. UCP1 is elevated in all of the fat depots. The exception is rapamycin, and that had already been published for reasons that are probably unrelated to any anti-aging effect that it has. Rapamycin had previously been shown to push down UCP1 in fat, and by gosh, it's true. So because rapamycin itself has a direct effect on UCP1 in fat, we can't really give credit or take credit away for that failure. But the other drugs, all of them elevate UCP1. Now let's look at macrophages in the fat. If anyone here is a macrophage biologist, you're going to hate me because I'm going to pretend that I only know about 2 kinds of macrophages. The M1 macrophage, which is pro-inflammatory, and the M2 macrophages. And I know all of your labs are devoted to complicating that story in productive ways, but I'm going to pretend I don't know any of that. We're going to talk about M1 pro-inflammatory macrophages and M2 anti-inflammatory macrophages, and please don't hate me. Hey, look, we're using ARG1, arginase, as our index for the M2. The original paper, we looked at 6 other things that are also present in M2. I'm just going to show you the ARG1 data. And for our pro-inflammatory macrophages, we're going to use nitric oxide synthetase. We've also looked at 3 cytokines in the M1, produced by M1 cells. And here you can see in the brown fat, inguinal fat, and perigonadal fat, this is wild type versus growth hormone receptor knockout. The anti-inflammatory macrophages come up and the pro-inflammatory macrophages go down. And if you look at cytokine production, the ones from the pro-inflammatory macrophages go down. If you stain sections and you look at surface markers, you see the same pattern. It's really true. That's true for Snell-Dwarf as well. And we've published it, and I won't show it to you, Amesdorf and PAP-A knockout as well. Now we can look at the M1 cytokines in the fat tissue. These are produced, the ones we're looking at here are produced by the M1 macrophages, and all of them are down in the Gurkha mice and the Snell-Dwarf mice in brown and both kinds of inguinal white fat. Consistent with the data I just showed you on the INOS story. And now we can look at the drug-treated mice and the calorie-restricted mice. So here are the M2 cells, here are the M1 cells, and again, every one of these is elevated in the slow-aging mice. We also have data on the cytokines, which is quite consistent with this. So the fat story is UCP1 is up, pro-inflammatory macrophages down, inflammatory cytokines down in all 10 models, and anti-inflammatory macrophages are up. Which is cause and which is effect is not clear yet from the fat tissue. Now let's look at the muscle. It's known, well, it's controversial, but some people believe, and I bet they're right, that these changes in the fat, in particular UCP1, are actually mediated by a small peptide called irisin. Irisin is a cleavage product of a muscle protein called FNDC5. FNDC5 can be clipped, the extracellular moiety, irisin goes through the blood and does good things to fat. It's controversial for reasons that I don't want to go into right now. Our data certainly fit that model quite nicely. So since we've seen these UCP1 things and the macrophage turnover in the fat, we predicted that irisin would be up in the slow-aging mice and that its precursor in the muscle, FNDC5, would also be up. And this turns out to be correct. So here's Snell-Dwarf, here's Gurkha mice. This is the plasma irisin, nicely elevated in both mutants. Oh, this is the relative protein expression. That is also changed. And this is plasma, right? This is FNDC5 in muscle. I didn't get it over to the right-hand column. So the irisin data and its precursor protein in the muscle tissue by Western blot show parallel changes, as we would hope. Many people don't like irisin assays by ELISA in serum. They've been plagued with problems. So I would not have believed this, except that it's completely congruent with the FNDC5 Western blots, which are relatively unambiguous. P10 also shows this. And now let's look at the drugs and the diet. Once again, they all show an increase in FNDC5 in the muscle, that's shown here, and irisin in the plasma. Interestingly, that's these little red circles. Two of the drugs, canagliflozin and 17-alpha-estradiol, show a sex-specific effect. They work in males only. And of course, you remember, those are the two that have a lifespan benefit for males only. So this is one of those instances in which the lifespan benefit, which is sexually dimorphic, is at least paralleled by a sexually dimorphic change in these biochemical indices as well. And the last thing I want to talk about is CAP independent translation. I gave a whole seminar on this yesterday. So for those of you who are at that seminar, you can go through the next few slides if you would like for me. This is the intro slide. Most RNAs, of course, are translated by a ribosome glomming onto the cap at the 5' end. It bumbles down the message until it gets the AUG start site and makes translation. But if you block that, if you block cap-dependent translation, a small subset of mRNAs, perhaps a few dozen, perhaps a few hundred, you can get that protein translated anyway. There's a 6-methyladenosine residue in the 5' untranslated region, and the 40S ribosome can find that if it's coated with the right proteins, YTHDF1 and YTHDF2. Well, Gonzalez, and then it makes this new subset of proteins. So this is molding the proteome by selective translation, not by transcription change at all, but by translation. So Gonzalo had published that this is elevated in Snell dwarf, Ames dwarf, PAP-A knockout, and growth hormone receptor knockout. 48 hours after you put rapamycin on a cell, they're churning out cap-independent proteins. 7 days after you give rapamycin to a mouse, they're churning out cap-independent proteins. And these proteins are cool ones. Some of them are involved in DNA repair, some in mitochondrial protection and production. Some of them are feedback, positive feedback loop. Now, the interesting thing from my perspective today is that GPLD1 turns out to be a cap-independent protein. So that links this selective mRNA translation story to the story I was just showing you about GPLD1 and its effect on brain. Let me show you a little bit of that. So here are two proteins, NDRG1 and MGMT. They're involved in DNA repair. And you can see in the liver, the muscle, and the kidney, they're elevated in Snell dwarf mice. They're also elevated in growth hormone receptor knockout mice. And when Gonzalo looked at the mRNA, there was no change in the mRNA. It's a protein change without a change in the mRNA. That's the first clue that it's translational control, selective translation. Okay, now let's go to the drugs, acarbose and 17-alpha-estradiol. When you use acarbose, which extends lifespan in both sexes, it also produces cap-independent translation of mRNAs in both sexes. So the black bar and the white bar, that's young and old. You can see that production of cap-independent proteins goes down with age. But in the males, that's what the red star means. It's retained at youthful levels. Whether you start the males 16 months before you kill them, these are being killed at 22 months of age, or even at 4 months before you kill them. 4 months is just enough. In some cases, actually, the amount of the cap-independent protein is way above young, so it sort of overshoots the youthful amount. But 17-alpha-estradiol does exactly that in the males and doesn't do it in the females. So this drug-induced switch to cap-independent translation of messenger RNAs for 17-alpha-estradiol is male-specific, but not seen. Here's an exception to the rule. Not seen for the sex-specific drug. And canagliflozin, Gonzalo has just submitted for publication. It's just like this. The males do it, the females don't do it. Since I want to leave some time for questions, do I want to do that? Let me do one more slide. So this is a MAP kinase pathway, and I'm going to do this too fast. There are two different MAP kinase pathways that Gonzalo has studied. One gets signals from insulin and turns protein translation on or off. That's this guy. And that is changed in all these mutant mice and by all the drug-treated mice. And the drugs are sex-specific. I won't walk you through all of the data, but that's the take-home message from that. This particular MAP kinase cascade shows changes that are completely consonant with the lifespan effects. It's true for the first kinase in the pathway, the second, which is the famous ERK1 and 2, the third kinase, and then for the eventual target, which is the translation regulator EIF4E. There's a second MAP kinase pathway in the paper, not in my slide deck, which does something entirely different. It sees inflammatory signals and it turns on acute phase proteins to try to buffer inflammation. That kinase pathway is also changed by aging, and that age change is affected by all of the drugs and by the mutant mice. But interestingly, it is not sex specific. That is, canagliflozin, acarbose, and 17-alpha-estradiol change the age effect on that pathway in both sexes. That's why we think, that's why I'm showing you this one. I think this is the one that counts. I think this one, the control of translation biology, is one of the ways in which these drugs lead to extended longevity. So this is the summary. There's a review article that just appeared in the new journal, Aging Biology, and this is part of that review article. This is one of the shock and awe slides where you're not expected to memorize it, but it's just to impress upon you that the data set so far includes multiple tissues. It includes multiple molecular as well as biochemical changes. And these are the models in which it is changed. The asterisk means it's sex specific and only the males do it. So I'm going to finish up by showing you the themes I'm trying to emphasize. The first theme is that aging rate indicators are not biomarkers of aging. A biomarker of aging is something that goes up with aging. So if it goes up slowly, then maybe you're aging more slowly. That's like an odometer in your car. It tells you how far your car has gone. This is quite different, unrelated to that completely. These are speedometers. They tell you at the time you take your tissue sample how rapidly the mouse is aging, because it distinguishes slow aging mice from regular old mice. It does not matter whether they change with age or not. Whenever we send in a paper, half the reviewers say, oh, wait, you haven't studied in old mice. You don't even know if it changes with age. Ha ha, you're rejected. So then we have to send back a rebuttal saying, we really don't care if it changes with age. That's irrelevant to the points we're making here. And about half the time, the editor notices this and agrees with us, and the paper gets published. There's a stochastic process. This is a biomarker of aging. It goes up with age, but in a treated animal, which is aging slowly, it goes up more slowly. That means you can only study it in old mice. How extremely inconvenient. It also means you can't use this as a screening test for an anti-aging drug. So here's a hypothetical aging rate indicator. When you add the drug, it goes up more or less right away. More or less is important. We don't know if that's a few weeks, a few months. We really want to find out. But the thing is, it's a difference between the regular group and the treated group survives. It's not a change in slope. It's a change in set point. And you can do the same thing, even if the thing you're measuring actually goes up with age. The question is it always higher in the treated mice? Or if the thing goes down with age, the question is it consistently different in the treated mice? I should send a free copy of this slide along with every paper that we're sending in for review. So I've just said that, but I'm going to say it again in case you're ever asked to review a paper of mine. Aging rate indicators are not biomarkers of aging. They are not like an odometer. They're like a speedometer, and it doesn't matter whether they change with age or not. And the second general point, good for biologists, is that if you're limited to transcription data, and the world is now currently being flooded by transcription data, RNA-seq has opened a floodgate for this. And the people who are interested in where the epigenetic factor is binding and where the histone thingies are binding, and ATAC-seq and what's open to transcription, they're obsessed with transcription. But these mechanisms are non-transcriptional. We would have missed everything here if I'd looked at transcription. What Gonzalo and Gina did was look at CAP-independent translation and something I didn't talk about today, chaperone autophagy. They'd be missed completely by transcriptome studies. The last general point for biologists is that most of these are non-cell autonomous. That is, if you think you've got a monoculture, you're going to dump some drug on it, something good is going to happen. That may happen, but you'd miss all of this stuff. Because all of this are a change in one tissue that's produced by a change in the other tissue. The brain changes are being produced by the liver and fat making GPLD1. The fat changes are being produced by irisin, and there are others on the list here. You don't model this in single cell culture. Unfortunately, you have to have an actual real live mouse. Okay, I'd like to know what the mechanisms are upstream. That's the target. That's the main reason I live. I want to find out what is upstream of all of these. What is the control process that regulates all of these in common? Because that's where we want to focus our attention as drug producers. How do they lead downstream to changes in liver disease and heart disease and cancer disease and Alzheimer's disease? Those are pathology questions that these aging rate indicators help you solve. How long does it take the drug to flip the switch? If the answer is a few weeks, we can use this as a rapid screening test for anti-aging drug. Can you flip the switch in people my age? I'd really like to know that. And we need data on mice to answer that. And since I want to leave some time for questions, I will skip the advertisement for the ITP. If you're interested in the ITP, either as proposing drugs for us or getting tissues from these mice, just let me know, and I will be glad to help you through that. So whenever I used to tell people I worked on anti-aging drugs, my wife was an English professor, I would be then roundly criticized. How dare you extend lifespan? There are too many old people, and I don't want to be like my 100-year-old Aunt Maisie. She's sick and tired and grumpy all the time. And making a lot of grouchy old people is not the goal of anti-aging research. In fact, none of our drugs do this. The goal of aging research is to create a brand new kind of old person. This is your typical average randomly selected 70-year-old. And if these drugs worked in people to the same extent that they work in mice, this would be the portrait of your randomly selected average 98-year-old person. All of and this is not just a fantasy or just a joke. All of our drugs do that. So I'm not making it up. They don't do it in people. They do it in mice. So that's the goal of aging research. And my wife's humanities professors who view this as a science is only bad for you. Science does only really, really, really bad things. I need to make sure they understand this is our goal. So those are the two talks I wanted to give, and I'd be delighted to have any questions. The people who are going to ask questions half an hour ago get to go first.